So we're going to be transitioning from talking about how proteins get secreted or targeted to membranes or various organelles to talking about how cells take up materials and how these two processes are related. So by now you should be comfortable talking about mechanisms of protein synthesis, so how it starts, what signals proteins have that direct the cell where to send that protein. You should be pretty comfortable talking about the synthetic role of the endomembrane system. So this means how proteins, for example, are targeted to the endoplasmic reticulum, what modifications they get there, how they're then transported to the Golgi, what modifications they receive in the Golgi, and then how they get sent to the membrane. And we'll be talking about the details of vesicle formation in the next couple of lectures. You should be familiar with and become comfortable talking about the different signals that are embedded in the sequence of a protein. So remember, these signals are part of the primary structure of the protein or the amino acid sequence, and there are very specific signals that target proteins to the endoplasmic reticulum, to the mitochondria, to the chloroplasts, and then other signals that target the protein to the nucleus or that tell the endoplasmic reticulum to add an N-linked sugar or the Golgi to add an O-linked sugar. So these are concepts and details that you should be working on becoming more familiar with. And we're going to start talking more about the roles of receptors and enzymes that act after signals are bound in this system. So, for example, at the side of the Golgi where secreted proteins are, there are specific receptors there that help sort the proteins and then help make the vesicles that will then go on to be secreted or head to the lysosome. And we'll be talking about the lysosome system, so the degradative role of this endomembrane system. So how cells digest mitochondria that might be damaged and be bad for the cell to keep around, how single cellular organisms can phagocytose material and degrade that material and use it as their food, those types of cellular behaviors. So just a reminder that until now we've talked about how proteins can be transported through nuclear pores and the characteristics of those pores that allow them to be a regulated gate for larger molecules to go in and out of the nucleus. We've talked about transport across the membrane of the chloroplast to mitochondria and endoplasmic reticulum. Just know that the peroxisome is another membrane-bound organelle that has a similar mode of import for the proteins that act there. And then we've started talking about transport by vesicles, particularly for proteins that need to be on the cell membrane or for proteins that need to be secreted like insulin. And these processes can be constitutive or unregulated exocytosis. So this is a way in which you now know that small amounts of phospholipid membrane can be added to the surface of a cell, for example. Regulated exocytosis, we know how neurotransmitters are held in wait at the synapse of a neuron and how then the cell knows when to secrete those neurotransmitters. And so now we'll start talking about how cells take things up. So when we first discussed membrane function earlier in the semester, we talked about a few ways of endocytosis that a cell participates in. And so at the time, we just talked about know that these are a few different ways, and now we'll talk a little bit more about the purpose and the mechanisms of these forms of taking up materials. So these terms should sound familiar to you. So phagocytosis, when a cell takes up a large particle that you can see, and that large particle then enters a membrane-bound vesicle in the cell. Penocytosis, which is also known as cellular drinking, which just takes up water without any selection, and it'll take up any solutes at the same time as indicated by here by this blue box into vesicles in the cell. And then we'll be talking in detail about receptor-mediated endocytosis. So this is a way in which cells can take up things in a more concentrated fashion using receptors. So again, penocytosis is how cells take up small vesicles. This will turn over small amounts of the membrane, which, as you know, also is replaced by constitutive vesicle fusion with the cell membrane, and it ingests small molecules that are in the extracellular space. So then there are a few ways by which the cell performs bulk transport that are going to be diagrammed here. So the first is endocytosis, and we'll talk about how this is receptor mediated. And when these vesicles enter the cell, what happens is the pH in the vesicles drops a little bit, and they're now considered an endosome. So prior to they were a transport vesicle, you get this drop in pH, and then they are characterized as an endosome. Early endosomes are a place where materials can be recycled and sent back to the membrane if necessary, and we'll talk about an example of that. If the pH drops further, then they're characterized as a late endosome, as you see here. And then those late endosomes fuse with lysosomes where further digestion of the material inside can happen. Phagocytosis, similarly, you have this large particle that is enclosed in the cell membrane. Now it's called a phagosome, and that phagosome will also fuse with the lysosome so that the material inside can be digested. And sometimes there are organelles in the cell that are damaged and unhealthy. So it's a really important process, for example, to turn over mitochondria that are no longer functioning correctly. And the cell does this by autophagy. So auto meaning self, and phagy eating. So it'll generate a membrane around the damaged organelle, and that autophagosome will fuse with lysosomes in order for the material inside to be digested. So this is an overall schematic of the GIRL endomembrane system. So we have our endoplasmic reticulum that is continuous with a nuclear envelope. We have the vesicles going from the ER to the Golgi. And from the Golgi, remember that proteins that are destined for the lysosome have a high mannose added in the endoplasmic reticulum. And that high mannose is a N-linked sugar. And that mannose can be modified in the Golgi. It's phosphorylated. And then it is termed a pi mannose, the PI, short for the inorganic phosphate, that is added to the mannose. That targets that enzyme to go into the endosome system. So for example, the enzymes that work in the lysosome have that pi mannose tag so that they get sorted to the correct vesicles to be sent to the lysosome. In contrast, transport vesicles have other receptors that allow vesicle formation for transport rather than trafficking through to the lysosome system. And similarly, when you have materials taken up, they enter this early endosome. That early endosome, the pH can drop and/or it can fuse with the late endosome for digestion of that material in a lysosome down the road. So endosomal sorting, as you may have picked up, happens at this early endosome stage, and there are other ways in which endosome vesicles help the cell function. So you may remember that I mentioned the default for proteins that are membrane bound in a polarized cell is for them to be sent to the apical plasma membrane here. However, as you know from the example of transport in a gut cell, we may want perhaps a glucose channel to be on the basolateral side of the membrane. So if that glucose channel is originally sent to the apical plasma membrane, because that's the default, the cell then takes that channel up into a transport vesicle and transcytoses it to the basolateral side. Similarly, at the early endosome stage, there's no digestion occurring, so materials can be recycled back to the membrane here, or they can be sent for degradation through a late endosome into the lysosome.